Unveiling the secrets of the coelacanth, a living fossil's anatomy. Imagine a creature that has remained largely unchanged for over 65 million years. A true living fossil that holds the secrets of our ancient past. The African coelacanth, Latimeria chelune, is just that, and its remarkable story is about to take a dramatic turn. For decades, scientists have studied this deep-sea relic, assuming its anatomy was well documented. But a new study reveals that long-standing descriptions of its head muscles were riddled with errors. This groundbreaking research not only corrects these mistakes, but also reshapes our understanding of how jawed vertebrates learn to bite, breathe, and swallow. But here's where it gets fascinating. The corrected map of the coelacanth's muscles makes it even more similar to cartilaginous fish, such as sharks and rays, and tetrapods, including birds, mammals, amphibians, and reptiles, than previously thought. The research team, led by Alessio De Tovo, a professor at the University of São Paulo's Museum of Zoology, re-examined two rare museum specimens and discovered that a staggering 87% of the previously identified evolutionary muscle novelties for the largest vertebrate lineages were incorrect. What earlier anatomists called muscles in the buccopharyngeal cavity turned out to be ligaments, structures incapable of contraction. This revelation has significant implications for our understanding of the evolution of suction feeding, a crucial skill that helped ray-finned fish diversify into half of all living vertebrate species. The coelacanth, lungfish, and most sharks, on the other hand, rely on biting. And this is the part most people miss. The evolution of suction feeding occurred at least 30 million years later in the common ancestor of living ray-finned fish. The story of how the research team secured the rare deep-sea fish is a testament to their dedication and perseverance. Datovo and his co-author, G. David Johnson, spent years persuading institutions to lend them the precious specimens. The Field Museum in Chicago and the Virginia Institute of Marine Science each entrusted them with a fish, which Datovo then painstakingly dissected over 6 months, preserving each element for future study. The team's findings not only correct 70 years of errors, but also identify 9 completely new evolutionary transformations in cranial muscles tied to feeding and respiration across early vertebrates. By rebuilding ancient muscle maps, the researchers used micro-CT scans to slot the correct muscles into place, tracing how jaw and throat mechanics evolved in the first jawed vertebrates. The coelacanth's unique position near the base of the lobe fin line that gave rise to all amphibians, reptiles, birds, and mammals means that these revisions ripple through the entire vertebrate family tree. The study is also an homage to Johnson, described by De Tovo as probably the greatest fish anatomist of his time, who sadly passed away in November 2024 while the manuscript was under review. The coelacanth's remarkable stability, confirmed by a 2013 genomic study, creates a living window onto ancient body plans, offering modern insights into the evolution of ray-finned fish and the traits that lobe fins passed on to terrestrial tetrapods. As De Tovo's team plans to compare the revised coelacanth anatomy with muscles in amphibians and reptiles, they invite us to ponder the significance of their findings. What other secrets lie hidden in the anatomy of these ancient creatures, waiting to be uncovered? The study, published in the journal Science Advances, is a powerful reminder that even the most iconic species can still hold surprises, and that the pursuit of knowledge is a never-ending journey. But here's the question. What do you think is the most significant implication of this study, and how do you think it will impact our understanding of evolutionary biology? Share your thoughts in the comments below. Coelacanth shocked scientists. 70-year-old evolutionary myth busted. Living fossil revealed. A discovery that turns decades of evolutionary theory on its head. Imagine a creature that has witnessed the rise and fall of dinosaurs and yet remains almost unchanged over millions of years. The coelacanth, a fish often dubbed a living fossil, has just provided groundbreaking insights that challenge long-held beliefs about vertebrate evolution. This revelation comes from a meticulous study by researchers at the University of São Paulo and the Smithsonian Institution, recently published in Science Advances. The team focused on the skull musculature of the African coelacanth, Latimeria chalumnae, and found startling inaccuracies in previous evolutionary studies. Only 13% of the muscle developments previously attributed to major vertebrate groups held up under scrutiny. Instead, they identified 9 new evolutionary changes that significantly impact how these creatures feed and breathe. But here's where it gets controversial. The coelacanth is more similar to cartilaginous fish like sharks and tetrapods, including birds and mammals, than previously believed. It markedly differs from ray-finned fish, which represent about half of all vertebrate species. This finding, led by Alessio Gatovo of USP, suggests a different evolutionary path from what was once assumed. One of the most crucial corrections made by the researchers involved structures once thought to be muscles in the coelacanth's head. These are actually ligaments, which, unlike muscles, do not contract. This error correction implies that previous models of how coelacanths and potentially other related species feed and breathe might be fundamentally flawed. The divergence of ray-finned and lobe-finned fish, which include coelacanths, from a common ancestor around 420 million years ago, marks a significant evolutionary split. Ray-finned fish, like those seen in home aquariums, use their mouth structure to create suction to ingest food, a trait that has given them an evolutionary edge. This study reveals that such adaptations arose 30 million years after their evolutionary paths diverged, not in their last common ancestor as once thought. And this is the part most people miss. The implications of these findings extend beyond just correcting past errors. They reshape our understanding of the evolutionary narrative of all large vertebrate groups, including humans. Coelacanths themselves are fascinating due to their rarity and ancient lineage. Found primarily in deep-sea habitats, these fish have changed little since the age of dinosaurs, likely due to their isolated and predator-scarce environments. This slow pace of genomic changes was highlighted in a 2013 study published in Nature. The recent study not only corrects historical inaccuracies, but also preserves these rare specimens for future research. Through careful dissection, which took 6 months, Gatovo was able to sepimate and preserve the skull bones and muscles for further study without the need for additional specimens. This research not only illuminates the evolutionary history of jawed vertebrates, but also sets the stage for future explorations into how these findings relate to other groups, including tetrapods. The use of advanced imaging techniques like 3-dimensional microtomography has opened new doors for understanding the ancient origins of vertebrate anatomy. What do you think about these findings? Do they challenge your view of evolutionary history? Could there be other living fossils out there that hold keys to the past? Share your thoughts and join the conversation below. 400-million-year-old fish changes everything we know about human evolution. Imagine finding out that a creature we thought we understood so well has been hiding crucial secrets about our own origins. A 400-million-year-old fish, known as the coelacanth, is forcing scientists to rethink key aspects of human evolution. But here's where it gets controversial. What we believed about this ancient fish's anatomy for nearly 7 decades might have been fundamentally flawed. What's so special about the coelacanth? The coelacanth, pronounced coelacanth, is often referred to as a living fossil because its physical characteristics have remained almost unchanged for millions of years. It resides in the deep ocean, hiding in underwater caves, and was once thought to be extinct until a live specimen was discovered in 1938. Due to its ancient lineage and striking resemblance to prehistoric creatures, scientists long assumed that studying the coelacanth could provide valuable insights into the early stages of vertebrate evolution, particularly how our ancestors developed the ability to eat and breathe using jaws. The big mistake: muscles versus ligaments. A recent study conducted by researchers from the University of Sao Paulo in Brazil and the Smithsonian Institution in the United States has challenged long-held beliefs about the coelacanth's anatomy. Published in the journal Science Advances on July 29, 2025, the study involved meticulous dissection of coelacanth heads using highly precise techniques. What they found was astonishing. Many of the structures that were previously identified as muscles were actually ligaments. This distinction is crucial because muscles enable movement, whereas ligaments primarily connect bones and transmit force. The discovery means that the coelacanth's capacity for jaw movement and respiration is much more limited than previously thought. Why it matters to us? So why does the anatomy of a deep-sea fish matter to our understanding of human evolution? The coelacanth belongs to the same ancestral group of fish that eventually evolved into all four-limbed animals, including mammals, birds, reptiles, and amphibians. This makes it a vital piece in the puzzle of our evolutionary journey. Scientists previously believed that certain muscles responsible for suction feeding, quickly opening the mouth to draw in prey, were present in coelacanths. If true, this would imply that these muscles evolved early and were passed down through subsequent animal lineages. However, the new findings indicate that these muscles were never there and likely emerged much later in a different group of fish. This shift in understanding could significantly alter the timeline of evolutionary development. Correcting 70 years of mistakes, the research published in Science Advances involved dissecting rare coelacanth specimens from museum collections in the United States. Over several months, the team meticulously separated each part of the skull and muscle system, confirming that 11 structures previously classified as muscles were actually other types of tissue. This means that numerous anatomy textbooks and scientific papers from the past 7 decades may require revision. In addition to correcting these errors, the study uncovered new features in the coelacanth skull that had never been described before. These discoveries enhance our comprehension of how early vertebrates developed their skeletal structures. Looking ahead, rewriting the evolution textbook. This groundbreaking research doesn't just fix old mistakes. It provides scientists with new tools to investigate the evolution of skulls, jaws, and respiratory systems in early vertebrates. It also sheds light on how tetrapods, the group that includes humans, began to diverge from their aquatic ancestors. By comparing 3D scans of other ancient fish skulls, researchers hope to construct a more accurate picture of our distant past. Future studies might explore how amphibians, reptiles, and mammals inherited or evolved away from these early anatomical traits. The study was led by Brazilian researcher Alessio de Tovo, with contributions from the late G. David Johnson, a renowned fish anatomist who unfortunately passed away before the paper was published. Their work underscores the ongoing nature of scientific discovery, reminding us that even after centuries of exploration, nature continues to hold mysteries waiting to be uncovered. By revisiting the past, we can gain new perspectives on our evolutionary journey. Source: this research was funded by the Fundação de Amparo à Pesquisa do Estado de São Paulo, FAPESP, and published in Science Advances on July 29, 2025.